We're going to change tumour slightly. I'm an esophageal gastric surgeon and also do research into the tumour microenvironment. And if you were at the Moynihan session yesterday, you will have heard John present an excellent piece of work, which hopefully wins, that I've collaborated with. More of that later. So, if you don't know, esophageal adenocarcinoma, in the UK we have the highest incidence of anywhere in the world. That's our dubious claim to fame. That's us in dark blue. We're only matched by Holland. Maybe we have something genetically similar to the Dutch. I'm not certain. But this is a disease that is predominant in the UK. We know that the only curative treatment, if patients are going to be cured, is an esophagectomy, with all the attendant risk that that delivers. We also know that there is some benefit from delivering neoadjuvant chemotherapy, and the current paradigm for treatment of esophageal cancer is intense staging, neoadjuvant chemo or chemoradiotherapy to a growing extent, followed by a treatment holiday, and then an operation. Clearly that is demanding of our patients, and only 1/3 of them are suitable for curative intent at the time of diagnosis. You can do a rough calculation based on a number of the large-scale trials, OEO2, MAGIC, and the like, and see that the majority of patients really don't benefit from chemotherapy. The number needed to treat to get one extra survivor is somewhere in excess of 11. If you can get the raw data, it would probably be even higher. Yet we give chemotherapy to nearly all of our patients. And in collaboration with 6 other centres around the UK, this is some very, very preliminary data. It's clear that you can divide patients based on response to chemotherapy into 2 groups. Those that respond, they are the lucky few, and those that clearly do not respond, and their outcomes are appalling. This is governed by the biology of the disease that we treat. Esophageal adenocarcinoma is believed to originate from Barrett's esophagus through a progression of low-grade and high-grade dysplasia to invasive carcinoma. This is a metaplastic cancer. The normal esophagus is not lined by columnar epithelium. It is squamous in origin. So something is going on that changes the cell type in the esophagus, an inflammatory process that may affect the epithelium and the microenvironment. So we've got better at understanding cancer cells. And one way that we've got better at understanding cancer cells is by the delivery of the Human Genome Project. Now, the first human genome was sequenced in 2000 and something or other, and it cost $1 billion and a bit and took 10 years of effort. A whole human genome can now be sequenced inside of a week and for under £1,000. That has revolutionized the way that we can understand cancer at the genetic level. The United Kingdom is leading, and surgeons are integral to us delivering samples to lead the world in defining the genetic landscape of esophageal cancer via the Occam's ICGC consortium, and there are people in the room who are contributing to that. And this is beginning to define potential new treatment groups in esophageal cancer purely based on the cancer cells. This is very early, unpublished data that suggests there may be 3 different categories of esophageal cancer, not based on anatomical location or CVUT classification, which are rubbish, but based on something biological about the tumour. More importantly, perhaps, these studies are telling us something about the origins of the disease. In the first paper published in Nature Genetics, we showed that driver genes are mutated early in the course. And when I mean early in the course, the majority of driver changes are found in Barrett's epithelium, and they're found in Barrett's epithelium that never goes on to become cancer. So if the building blocks are present at the beginning, something else is determining the fate of those cells. And I use that as an excuse to say that drivers of disease progression lie beyond mutations in cancer cells. But I think, having heard Alex in the first talk, we're beginning to build a picture that maybe it's not just all about the cancer cell. So you've heard that an esophageal cancer, just like a colorectal cancer, is not made up of one cell type. These are complex organs that involve fibroblasts, immune cells, lymphatics, and blood vessels, and other cell types, and they all interact with each other. I'm going to focus initially on the cancer-associated fibroblast. So fibroblasts in all of us make the architecture of our tissues and have a really important role in wound healing. If you were to cut yourself, the fibroblast in the extracellular matrix reacts to close your wound. It produces new extracellular matrix. It is a secretory signaling cell, and it's a contractile cell. So it binds the extracellular matrix and pulls it in to close the wound. And it secretes factors into the matrix to add that closure. It's an important cell for our biology. There is a process by which a resident quiescent fibroblast undergoes differentiation into a more muscle-like cell and takes on a myofibroblastic phenotype. And when it's become fully differentiated, this cell expresses a protein called smooth muscle actin that helps it anchor it to the extracellular matrix and become contractile. This is under the influence of TGF-beta, something clearly seen in the esophageal epithelium during tumour progression. This is important for some of the slides later. Differentiated myofibroblasts are SMA positive. So we looked at a cohort of patients who have had resections for esophageal cancer, and we stained their stroma for SMA. And you can clearly divide patients into those that have high or moderate levels of SMA, shown here, against those that have no SMA staining. And there is a clear split on Kaplan-Meier's. Those without a reactive stroma do better. And this is not just seen in esophageal cancer, it's also seen in colorectal cancer and head and neck cancer. You can stratify patients for survival based on their stroma. Interestingly, in the esophagus, if you do a multivariable logistic regression with this, you can show that alpha SMA positive stroma is more predictive than T, N, M stage, or positive resection margin for survival. Numbers are small, pinch of salt. We've also looked at whether this determines response to chemotherapy, and there is a direct correlation with expression or lack of expression of SMA and response to chemotherapy after resection. So not only can you give a prognostic detail for the patients, you can also learn something about their potential to respond to chemotherapy. So we can take these cells from the cancer, we can take these cells from normal esophagus. I say normal, it's being treated with chemotherapy, it's from a cancer patient, but it's distant from the cancer site, and we can compare NOFs, normal esophageal fibroblasts, to cancer-associated fibroblasts, CAFs. And you can show that the cancer-associated fibroblasts do indeed express high levels of SMA. They are myofibroblastic compared to their normal comparators. And they are mesenchymal. They come from the right place. We've also shown that these cells express SMA using immunofluorescence, and they are biologically active. They are contractile in that they will contract a collagen gel, and they will do that to a greater extent than their normal counterparts. So we've got some cells to play with in the laboratory. We've also shown that these CAFs promote cancer cell invasion just by themselves, using dual chamber assays where you use conditioned medium from the fibroblasts. The fibroblasts essentially have been living in it and secreting factors into it. We just then take the medium and use that as a chemoattractant, and compared to their normal fibroblast counterparts, the cancer cells invade to a much, much greater extent. And using the organotypic models that Alex described earlier, you can show exactly the same thing. The only difference in these two models is that one has normal fibroblasts in the bottom, one has cancer-associated fibroblasts in the bottom. And you can clearly see more invasion on the right-hand side. So these fibroblasts are determining the invasion of cancer cells. You've also grown animals and compared tumours that have no fibroblasts, normal fibroblasts, or cancer fibroblasts in them, and the tumours are bigger and the animals die earlier with CAFs. And that's not just restricted to historical cell lines. The problem with esophageal cancer biology is there are probably about 10 bona fide cell lines, all from about 1980. None of them I trust. So with a newly derived cancer cell line, exactly the same phenomenon is seen. Bigger tumours with CAFs, more invasion, and the hint of resistance to chemotherapy. So we've set up this model where I can probably convince myself that cancer cells under inflammation, under the drive of inflammation, in the lining of the esophagus become adenocarcinoma cells, there's also interplay with normal fibroblasts being differentiated to CAFs and interplay between the two. But what is in the middle here? An important question. What's the interplay between cancer fibroblasts and adenocarcinoma cells? And we have looked at a range of secreted molecules, one of which is a matricellular protein. It's secreted by CAFs into the extracellular matrix called periostin. And when you knock out periostin, if you prevent periostin expression, you have downregulation of invasion, both in dual-chamber assays and in our more physiological organotypics. The only difference here, these both have CAFs in them. The only difference here is that on the right, we have inhibited the production of periostin. So with a single gene downregulation, single protein downregulation, we can prevent invasion in this model system. Clearly, it won't be as simple as that in a patient, but we're developing a story where these CAFs are important. And that makes sense because binding of ligands in the extracellular matrix is known to have a range of downstream effects on cancer cells that end up with proliferation, survival, migration, and invasion. The matrix is important. We further went on to show that periostin can stratify patients for prognostically in a similar way to SMA, and that the periostin is secreted at the junction between stroma and cancer cell and comes from the cells that make SMA, the fibroblasts. So this is relevant in human tissues. So that's one example of looking at how mediators from cancer fibroblasts influence adenocarcinoma biology, in particular invasion. But we also know that they influence collagen in the extracellular matrix. Those of us that take tumours out know that some of them have a really dense desmoplastic reaction, can be hard as wood. And in the esophagus, that's particularly so. And that is caused by changes to the matrix delivered by fibroblasts. So Chris Hanley, who works in the lab alongside Gareth Thomas, Professor of Experimental Pathology in our group, we published this in OncoTarget last year that showed that if you use fancy scanning effectively, that's all I need to know about it, but you see aligned and elongated collagen fibers in cancer compared to the normal submucosa or in the squamous epithelium. These collagen fibers are longer, they are more structured, they are more aligned, and that means that the tumour cells can invade more easily. If you take this forward, you can stratify patients who are SMA positive with even more certainty if they have aligned and elongated collagen fibers. So we've got some evidence that cancer-associated fibroblasts influence cancers via mediators. We've got some evidence that they influence cancers via changing the matrix within which the cancer exists. But what about immune modulation? This is the sexy thing in cancer biology at the moment. Everyone wants to give someone an immune modulator. Are CAFs important? Well, this is immunology for surgeons, or at least for me. There are two arms to the immune system, the innate and adaptive immune systems, and the cells that we think or need to know about that are important effectively are CD8 cytotoxic T cells. These are T cells that are or can infiltrate tumour, recognize tumour, engulf tumour and destroy it. And CD8 cells are important for this process. So we simply looked at esophageal adenocarcinomas, and we looked across stage, and we looked at the number of T cells within each stage of tumour. And you can see that that goes down as tumour stage increases. There is a correlation between tumour stage and CD8 count, which is highly significant. Cause or effect. But if you stratify patients based on the number of CD8 positive cells in their cancer, there is a clear split between those who have a high CD8 infiltration and those who have a low CD8 infiltration. And this is mirrored in other tumours across solid organ malignancy. If you have a good immune response to your tumour, you will do better. And this is the principle of immunotherapy as we currently know it. Interestingly, we then did a logistic regression exercise looking at all the possible modifiers of prognosis, and unsurprisingly, in univariate analysis, T, N, M stage, invasion and resection clearance are positive, but so CD8 cells. And on a multivariate analysis, the only things that remain significant that predict prognosis are end-stage resection clearance and CD8 infiltration, showing the importance of the immune system to esophageal adenocarcinoma. Yet there are no immune modulators currently in clinical practice for esophageal cancer. Trials are coming. How does this all add up? And why am I telling you about this? Well, do CAFs prevent T-cell infiltration? In theory, they should, based on the changes to the collagen morphology. And we have some evidence that if you have high levels of SMA, you are more likely to have a lower CD8 infiltrate, an indication that there may be a correlation between the amount of SMA-positive cancer-associated fibroblasts and an inhibition of T-cell infiltration. So we've got these fibroblasts, they mediate things to the cancer, they change the collagen matrix, they might have an impact on the immune system. What about drugging them? And where could we drug them? So we could drug them here, we could drug them here, we could use immune modulators, or what about going back to the source, drugging the cancer fibroblast, and then taking out all three? Is that even possible? Why on earth am I standing here showing you this slide? Well, I think it is possible, and this may be the class of drug that makes it possible. So, those of you who know anything about Viagra, and I'm sure there's no one in this room, will know that it works on nitric oxide pathways to decrease cyclic GMP in the cell. That's its primary mechanism. And by decreasing cyclic GMP, you have a variety of downstream effects. Look up Viagra if you don't know anything about it. I stumbled across a paper that showed in benign prostatic hypertrophy, Viagra-like analogues can de-differentiate activated fibroblasts. And I thought, well, let's see if that works in cancer. And I wouldn't be standing up here if I didn't think it did. So if you give Viagra, or this is Cialis actually, vardenafil, in large doses, you can completely inhibit alpha SMA expression in CAFs. This is quantified on this graph, and this is a three times a day dosing with vardenafil, knocks down alpha SMA expression by 80%. This is correlated with a decrease in invasion in transwell invasion assays. There is a decrease in the amount that the CAFs can contract a collagen gel in the presence of PDE5I, and, importantly, invasion is virtually abolished if CAFs are treated with a PDE5 inhibitor in organotypic models. We might have a drug. And I've blatantly stolen a slide from John's talk yesterday from the Moynihan session that shows that actually we might be able to use this as a deliverable drug in human cancers. There is better effect of standard of care chemotherapy agents when you give a PDE5 inhibitor in the presence of CAF. And we can make patients who may have never responded to conventional chemotherapy potentially respond. Clearly, this is patient-specific, and we will need to determine which patients we should give this drug to, but you can quickly see that this drug that's given in billions of doses around the world every year could be translated to a cancer therapy. So there we are. We potentially have a new drug that targets the source of mediators and collagen changes in esophageal adenocarcinoma that may improve survival in the patients that I treat. So in the future, where we're heading, bringing this all together, is using biopsies from diagnosis to stratify patients based on the phenotype and genotype of their cancer cells, but more importantly, modeling how their cancer cells interact with their tumor microenvironment in the laboratory while they receive induction chemotherapy, so we can then go on to deliver them tailored therapies that will actually work, rather than the blunderbuss that we currently give, which is platinum-based triplet chemotherapy plus a bit of X-rays, and see if it might help them. And this is why we're here. I really, really, really don't want to be doing this to people, although he saw his thumbs up day one after his esophagectomy. If we can get to a situation where we can treat this with a targeted therapy and avoid operating, then I have been a successful surgical oncologist. And as you might imagine, doing this sort of work involves hundreds and hundreds and hundreds of people and only a few of them are up there who I should thank. And for those that I haven't put up there, I thank you and sorry for forgetting you. Thanks for listening. Chairman, questions?
Any questions?
I have one. I have two. I might have missed it. What does periostin do? And when you treated with the PDE5 inhibitor, you showed that it took out SMA. Does it also take out periostin?
Okay. So periostin is a multifunctional protein, actually. It's secreted into the extracellular matrix. It binds integrins on the cell surface of epithelial cells and has a variety of effects. Autocrine, paracrine, a whole world of stuff. All sorts of things. And the second question, PDE5, does it knock down periostin? I don't know is the honest answer.
Okay.
Probably, but I haven't looked.
Okay.
Question for me, Tim. So as someone who has no experience of Viagra as well, can I ask, so have you thought about using it in a prophylactic setting in patients with Barrett's?
Yeah, I think it would be a harder trial to do, and you'd have to give people Viagra for a long time. It's clear that individual patients have different responses. We've got some data to suggest that some fibroblasts have a rebound expression of PDE5 when the inhibitor is withdrawn, more than others. And the doses we think we'd have to give would be 3 times a day dosing, the kind of doses that you would give in pulmonary hypertension or fibrotic disease, where it's currently licensed, not the occasional dosing as it's used recreationally.
It may be an easy trial to sell.
Yes, there is some amusing tales to tell about 66-year-old men, the average person with adenocarcinoma of the esophagus being given Viagra, but we can sell that trial, probably. The answer is going to be that it will work in multiple ways. And there's some really, really interesting evidence from coming out of Cambridge currently that suggests that CAFs actually can present antigen as well and can modulate T-cell trafficking, T-cell homing to tumour cells. So they're not just as simple as even I've described them. They have antigen modification, immune modification-like properties as well. So they do lots.
Tim? Sorry, what is the state of the fibroblasts in sort of the Barrett setting?
Good question. And the answer to that is, apart from expression profiling done in a couple of studies, no one actually knows.
Okay.
And we're currently looking at a variety of tissue samples taken across development of carcinoma from Barrett's through low and high grade to see where and when during that process there is a change in the stroma, because that's the pace where you could actually target to prevent tumour development.
Yeah.
Let's go, Hayley. Right, so our final speaker of the afternoon is Hayley Francis. Hayley is a senior staff scientist at the Wellcome Trust Sanger Institute. She undertook a PhD at Cardiff University investigating drug resistance in breast cancer before she moved to the Sanger as a postdoctoral fellow, and she spent the last 2.5 years in the lab of Matthew Garnett, investigating novel cell derivation technologies and determining their utility for cancer research and drug screening. And we are really, really honoured to have Hayley from the Sanger come and talk to us about the next generation of human cancer models. Thank you, Hayley.
Thank you. Thank you for inviting me. So as Tim said, a bit of a change of track, I think. So I'm going to talk to you about sort of new cancer models that I'm working on and how we want to make these for the research community and provide better models for labs such as Tim and Alex's to be using to do their research. So in Matthew Garnett's group at the Sanger Institute, for the last 7 years, we've dedicated our time to conducting high-throughput cancer cell line drug screens. So at the Sanger Institute, we hold one of the largest collections of cancer cell lines. We have about 1,001, and they cover the majority of cancer types. And as I said, we've spent our time running high-throughput drug screens where we want to model drug response. And so far, we've generated over 500,000 IC50 values. In addition to this, we've also genomically characterized all of our cell lines. We know all the genetic events that are happening within those models, with the goal of amalgamating that data together for therapeutic biomarker discovery. So we can identify genetic biomarkers that determine whether patients will respond or not to given therapies. And at the bottom of this slide, I've just shown some of the associations that we found within our drug screen. So, like many others within the community, we utilize cancer cell lines heavily. They're easily to use experimentally, and we use them above and beyond the drug screen. However, we are completely aware of the inadequacies of our current cell line collection. So, importantly, they fail to reflect tissue tumor architecture. We grow them as 2D monolayers on a plastic flask. We all know that that's not what a tumor looks like in the body. There's a poor success rate of derivation. So generally when a sample is taken down to single cells, digested into single cells and put onto plastic, only the most aggressive clone will grow. And generally only the most aggressive cancers or tumors will grow. So our cell line collection is completely biased towards more aggressive cancers. They've also adapted to culture, so they represent only a subset of the cells which favor 2D growth. So just like I was saying, only 1 or 2 clones will grow out. There's a poor representation of some cancer types. So Tim already alluded that there's only 10 esophageal cancer cell lines that we can use. There's probably a similar number for prostate as well. There's little or no associated clinical data, so we don't really know the sample history of these cell lines, which can make them difficult to use. And importantly, for the work that we're doing in Matthew Garnett's group, they fail to encompass the genetic breadth of some tumor types. So if we're using a collection of models that fail to cover the genetic breadth of cancer, how are we supposed to identify biomarkers of drug response? Therefore, we've always been very excited about new sort of novel cell derivation technologies, and in particular the organoid technology that Alex has already introduced in this session. So I just thought I'd tell you a little bit more about organoids as the rest of my talk focuses on organoid technology. So organoids now allow us to grow both normal and diseased tissue with a very high success rate, much better than we were ever able to do in the past. We culture them in specialized conditions to mimic the in vivo environment. And by doing this, it prompts signals that allow these organoids to become self-organoized 3D models, and they have multiple cell types, including stem cells. So the seminal paper for organoid technology from Hans Clavers' group was in 2009, as Alex already said, and it was looking at colon tissue. And what Clavers was able to show was that you could take a single crypt from a patient. So here you can see the stem cells reside at the bottom, you have the progenitor cells, and they differentiate up. And if you take a single crypt or even a single stem cell and you culture them in these specialized conditions, what you form is what he coined mini guts. So they are able to self-organize into the crypt structure. So you see the stem cells at the bottom, they differentiate up, and they form these spheroid structures, and they're mini guts. And this is what they look like in the lab under the microscope. So just to show you how we make these, people like Tim give me tissue samples, both normal and cancer. We literally mince and digest them just as we used to do for the 2D cell lines. But instead now what we do, we resuspend them in something called BME2, which is like Matrigel. So what Matrigel is liquid at 4 degrees, but above 10 degrees it begins to polymerize and forms like a solid structure. And it's basically made up of collagen and laminin, so it has a number of growth factors and so forth in it. And then we plate them in 6-well dishes in little 10 to 15 microliter bubbles, and the model and the organoids just grow inside. So you're preventing them from hitting the plastic, and you're providing a physical support for them to grow. So we grow them in 6-well plates. You can easily break these up, and each of the stem cells within these will grow on and propagate and make another organoid. So they're easily expanded to be used in drug screens and so forth. So this technology has now moved into a number of different tissue types. I've listed some here, and there are more. And there's a number of different applications and investigations that you can use them for. So as I said, at the Sanger, we're doing a lot of drug screening, different genomic analyses. Can we make new cell models that now cover the genetic breadth of cancers that we're interested in? Personalized treatment, gene editing, and then I guess going back to what Tim and Alex have been talking to, complex modeling. Could we use these new, better cell models, put things such as CAFs and immune cells in with them, and look at response to treatment? So following the seminal paper from Hans Clavers, we quickly established a collaboration with him to do a proof of concept study within colorectal cancer. And we made what we called an organoid biobank. So 20 patients were approached who were undergoing surgical resection of their colon tumours and agreed to allow us to take sort of surgical biopsies from their tumour as well as a matched normal piece. And we derived organoids both from the tumour and the normal. And we also took a part of the tumour and done immediate sequencing. So this would allow us to compare the genomics of the tumour to the genomics of the organoid. We expanded and stored them, and then we also completed DNA sequencing and drug screening on this panel. So one of the first things to note, just from H&E staining, one thing that really sort of blew us away, I guess, in some respects, was some of the tumour architecture was maintained in the organoid. So, for example, patient 19A, this tumour was very dense, the whole lumen structure had been lost, while for patient 8 and patient 17, you see some of the lumen structure is still there. And when you look at the matched organoid, that's exactly what we saw. So, 19A formed these really dense cystic structures, while patient 8 and 7, the lumen structures were still there. And then just going on to the genomic studies that we did. So this first graph on the left-hand side just shows mutational burdens. That's just the number of mutations that were in the biopsy and in the organoid. So it's quite difficult to see, but the B represents the biopsy and the O the organoid. You can see, for example, for patient 10, there's a very similar number of mutations both in the biopsy and the organoid, and this is the same for both. You can also see that the panel splits into two groups, hypermutated and non-hypermutated. Very well known that colorectal cancer, a subset of colorectal cancer patients are microsatellite unstable and have a heavy burden of mutations, which is what we saw here. This graph here shows the concordance of mutations between the tumour and the organoid. So the green shows concordance. And as you can see, over 80% of the mutations found in the tumour were captured by the organoid, which is something that we've never seen before. However, you still have some mutations that are in the organoid only or in the biopsy only. This suggests that maybe you are losing some clones, and this culture technology doesn't allow you to propagate all of the clones that you start with in a tumor sample. And it also suggests that if the organoids are continuing to mutate, that might be something different from what is happening in the cancer. And then lastly, we wanted to compare the frequency of recurrent mutations that we see in colorectal cancer with large clinical data sets. We did this by going to the Cancer Genome Atlas. So if you just concentrate on these genes here, so these are the top recurrently mutated genes in colorectal cancer, and we've compared them to TCGA. And you can see, so TCGA is in the light grey, and this cohort in the dark black. And we have a very good concordance with the reported alteration frequency from the TCGA. And this is only with 20 samples. So from the genomic analyses, we really did start to believe that organoid cultures were faithfully capturing the genomic features of the primary tumour. And even with a small panel of 20, we're beginning to capture much of the genomic diversity of colorectal cancer. So as I said, we're also performing drug screens. A little bit different to trying to do cancer cell line drug screens. So I've just popped this up here just to show you how we actually do it. I guess the main difference is we now put a layer of matrigel at the bottom and we see the organoids on the top. We have a drug library of about 700 compounds. Our drug library is mainly clinical, so we have most chemotherapeutics, clinical agents, preclinical compounds that are looking really interesting in clinical trials, and then a subset of experimental compounds that are hitting sort of very interesting targets. We then use a metabolic endpoint assay, so that just measures indirectly viability by looking at the number of cells that are still metabolically active, and then we're able to read this and just model drug response, which is shown here. We then amalgamated the sequencing data, so all the genomic data with the drug response data, and we wanted to look for known gene-drug interactions. So one such is KRAS status in response to cetuximab, an EGFR inhibitor. So cetuximab is an EGFR inhibitor. EGFR is a receptor tyrosine kinase that sits at the cell membrane. However, cetuximab is only active in patients that have a wild-type KRAS. If you have a mutated KRAS, this pathway is continually active regardless of EGFR inhibition. So each of these grey dots represents an organoid. And you can see that in the KRAS wild-type organoids, the cetuximab IC50 is much lower than in those that are altered. there were 2 organoids that didn't fit this bill, and you can see that one is BRAF V600E mutant, so that's a mutation of RAF, again downstream of EGFR, and a known resistance mechanism to EGFR inhibition. The culprit for resistance here is still unknown, and we're looking into that at the moment. And then secondly, another well-known gene-drug interaction is TP53 status and response to Nutlin3A. So Nutlin3A is an inhibitor of MDM2. MDM2 is a regulator of P53. However, so if you inhibit MDM2 with Nutlin, and in the wild-type P53 setting, this allows P53 to become active and initiate apoptosis and so forth. If P53 is already mutated, there's a loss of function and Nutlin has no effect. And this is exactly what we saw. Wild-type TP53 has a very low, that cohort has a very low IC50 to Nutlin, while those that altered are resistant. Again, there was one organoid that didn't fit the bill, and when we've done some P53 staining, we found P53 to be stabilized, which is shown by this dark brown stain in here. So this suggests that even though TP53 was wild type, the pathway was mutated. So in terms of next steps and future work, we're really excited about the utility of organoids and their potential for cancer research. So we've extended our collaboration with Hans Clavers with the Stand Up to Cancer Dream Team project. And this is looking at tumor organoids as a new preclinical model for drug sensitivity analysis. So we're deriving 300 organoids from 3 different cancer types. So we're continuing to expand on our work on colon, pancreas, and breast. And we've already started doing drug sensitivity screens to both the colon and the pancreas organoids, and have now moved not only for doing single-agent drug screening, but combinations as well. We've also been working with Rebecca Fitzgerald, also part of the Occam study that Tim was talking about earlier, to do some esophageal organoid derivation. As Tim's already alluded to, esophageal cancer is a cancer of high clinical unmet need, and there's not much research tools out there for people like Tim and Rebecca to use. So we've been able to already double the number of esophageal cancer cell lines that are available to the research community with this technology. We have matched tumor sequencing for the majority, again through the ICGC work and Occam's. And really, really nicely, we have all associated clinical data, including pathological reports and tumor response to neoadjuvant chemotherapy. So this can allow us to determine if the organoids are showing the same drug response as what the patient did when receiving chemotherapy. We're also doing some genomic stability studies. So this is looking about what happens when you're keeping these cultures in culture for a long time. Are they continuing to mutate? Do they change from the cancer that they came from? Does that mean we can still use them if they've been in culture for a long time? So to do this, we're extracting DNA at different ages and then doing whole genome sequencing and comparing to the primary tumor. We've also had the opportunity to derive organoids from patients who unfortunately died of advanced esophageal cancer. So we're taking multifocal biopsies from warm autopsies and deriving organoids from these. Initially, this was quite hard, as these patients, as you probably know, generally have systemic infections, and those infections come out with the biopsy. So a lot of our first samples, we just lost to severe infection. But the use of next-generation antibiotics has allowed us to turn this around. And for one patient, we have quite a few samples now that we're investigating. And then lastly, I guess the work that we're sort of moving on to now, and both Alex and Tim are involved in this, is working with CRUK to do a pilot study to determine what would happen. Could we make the next generation of cancer cell lines? Could we make thousands of organoids? Could we highly annotate them genomically, do drug sensitivity screens, and bring all of that data together, for multiple applications for the research community to access and to use, to also improve patient therapies? And would they be useful? Would they be better than what we currently have? So that's something that we're working towards at this moment in time. We're currently going to derive 250 from colon, esophagus, and pancreas. And we're working with various clinical sites in CRUK to do this and determine the utility. I guess one thing to note is just in terms of how well these models, I think the important thing for these models is to show that they are predictive of patient response. And there's a couple of clinical trials that are already ongoing from the Clavers lab, where he's doing co-clinical trials and taking samples from patients with advanced colorectal cancer undergoing chemotherapy. And he's derived organoids from these patients, and then he's comparing the response to chemotherapy in both to determine predictivity. And on that note, I would just like to thank everyone that's been involved, so all the patients who've allowed us to take samples, to be involved in all these different projects. Sania and everyone that was involved in the colon organoid paper, Rebecca for helping us set up all the esophageal derivation work, and all the clinical sites are involved in the new project.
I think a question is coming. Excellent. Saves me thinking of one.
Subject to simple surgeons like me, and I'm only sorry that many more of my colleagues weren't here to hear it. I'm sure they will be hearing from you in the future, and it'll be an important factor in how we manage our patients. That's not a question. The question is, Tim, you mentioned that adenocarcinoma of the esophagus is a British disease. Can you elaborate on that and tell us perhaps why?
It's not purely British disease, although we are we have the highest incidence. It's a Western disease, and it's something to do with the obesity, reflux, and our Western lifestyle. There must be some sort of genetic predisposition, because otherwise, why would it just be in the United Kingdom? You'd expect it to be in America at a higher incidence, although they do see a high incidence of esophageal adenocarcinoma. Other than that, we simply don't know. And we would hope that some of these studies will give us some of the answers.
Thank you.
John?
Interesting question. It's a good question. So the one thing I can say is I'm unable to derive organoids from normal esophageal, which is probably due to the squamous nature of it. And I think probably going back to Tim's diagram, where he showed when you're going through that inflammation, they show this more intestinal-like structure that I'm able to derive from esophageal cancer samples. So I think Clavers his work has always been quite nice, because he's always started from the normal setting and then translated that work from the normal setting into the cancer setting, and had that, I guess, the thought process that if it's going to work for a normal, then it should work for a cancer, because that's where the cancer started. Even though, as these guys were saying today, the microenvironment is also changing. So to be honest, I don't know. I think even within the cancer, we've go back to the hypothesis of cancer stem cells. I think they're still there. There's a population that are still there. And I think regardless of whatever stem cell that you look at, Wnt is always going to be important for those stem cell markers. So I think that's why they're there, even though we don't fully understand it as well as we understand the colon. The colon stem cell niche, is very niche, shall we say. The rate of turnover of the stem cells there allow you to look at it in much more detail than anywhere else, which is why he started there. But he's shown for all of those tumor types, liver, pancreas, that it's LGR5 positive stem cells that you take out and will form organoids. And regardless of the tissue type that you're looking at, you're manipulating the Wnt pathway by manipulating LGR5. Yes, so Clavers has done that in Barrett's and in esophageal, but he's just unable to derive at the highest success rate of colon. Yeah, so he's done it for esophageal cancer with us as well. It's true.
Sorry, go ahead.
Thanks for that. That was one of the most interesting pieces of science I've heard in a while. I don't hear much because I'm a clinician, but
Thank you.
If I may ask, it seems to me almost like we're fighting with tumour almost within an individual. It's an evolutionary battle against an accelerated evolution. There's so many different factors in each tumour in an individual. You take that tumour out, and then you can look at selection pressures in vitro or ex vivo, I should say. Do you think cancer treatment's going to become completely individualised in the future? It's almost a unique situation for a given person. Their tumour will develop in certain ways according to certain pressures, and you'll be able to monitor that in a lab.
Yeah, so I think if we're going to do it on a per-patient basis, we should start building all of those buildings on NHS clinical sites right now to start culturing cancer cells from every single patient. I think the mindset that we've taken at Sanya is that if we build collections that are large enough, that cover the clinical breadth of each of the cancers that we need to treat, all of them, that we will be able to find biomarkers of drug response and, I guess, particular genetic backgrounds that determine aggressiveness or non-aggressiveness. So that's what we've taken, but I think it's very true. I still like to think we'll be able to group them and that it's not going to become completely personable, but I think we definitely have to make that move towards personalized medicine to be able to better treat our patients.
Do you think it will be something that will be affordable in the future? Because I can see a situation where people have drug X and then they need, that doesn't work, they need drug Y, and then they need to throw in an angiogenesis inhibitor here and then an inhibitor of this, that and the other, intersusceptive inhibitor in, drug Z2, drug Z3. Could it become something that will be unfeasible, do you think?
Potentially. I think the advantage of working, so for me working at the Sanya Institute, we do everything at scale. And we're set up to do things at scale. So for example, generating large biobanks of thousands of organoids is not out of the realms of possibility, especially with collaborations with other people around the world, like Clavers, who's already generating hundreds of these things anyway. So I feel like it is feasible at the moment, and we can have a good stab at it to get to a certain point, but very true, it might become a point where to get to as personalized as we need to be to effectively treat all of our patients, it might not be feasible.
I can see in the NHS a lot of nice questions being asked.
Where we might have traction with that, to part answer that question, is that using these models, we may be able to determine the drivers of disease development and therefore major on prevention of disease and prevention of progression of disease rather than treatment of established disease in the longer term. And therefore, that's not as expensive as giving a cocktail of 20 drugs to one patient.
Yeah, I think that's an important point that Tim has said, that for traditional cell lines has always been the most aggressive tumors that we've been growing in the lab, while with organoid technologies, as I said, it's derived on normal cell growth. So, we're able to grow Barrett's organoids, we're able to grow adenomas, and therefore, we can really start investigating pre-neoplastic lesions and how to start them progressing into cancer.
Great, thank you.
Hayley. 2 questions, if I may. First one, have you seen any progression in your organoids from early stage to more later stage behavior of disease? So,
Good question.
So, for example, your Barrett's ones or your colorectal ones? Yeah,
so the Barrett's are an interesting one because our success rate is much lower. Not all of them grow. And a lot of our Barrett's have been taken with endoscopies, which is quite interesting as well, because we don't know if they are, from patients that are going to go on and develop cancer, or if those are not. So we don't know if there's a correlation there. In terms of the ones so for colon, we haven't grown any adenomas, but we don't see really them become more aggressive. So we try and propagate to drug screen as quickly as we possibly can, and then we repeat those drug screens months later, to see, I guess, looking at phenotypic changes of drug response, and they seem to stay the same. So I don't see changes in aggression, shall we say, but obviously they must still be genetically changing, especially, those colorectal organoids that are MSI. Thank you.
And my second question, going back to my analogy with the burgers, because it's getting near to dinner time and I'm feeling a bit peckish. So the burger was quite complex, but here we're sort of reducing it again to only 2 of the components, so the burger and the bun. Given some of the metrics that we're interested in, like drug response, heterogeneity, regression, etc., how can we take organoids further to try and model the complexity? Do you have any thoughts on that, please?
So I guess with respect to tumour heterogeneity, I think this model is fantastic for that. Like, we've been able to show that when you take a tumour sample, you take it down into single cells, many of those single cells will form organoids. So you're retaining polytonality. In terms of the simplicity of the model, again, very true. This is an epithelial model. We don't have CAFs, we don't have immune cells. But it's work that we're doing with Tim at the moment. So the organoid that we've derived with Tim, he's been able to extract the fibroblasts, both CAFs and normal, and we're working to put them back together and looking at effects of drug response when CAFs are there and CAFs are not there.
Thank you.
So no more questions from the floor. I would like to thank the very many of you who came to this session, and thank you for the fantastic questions, because they were absolutely brilliant questions. And I'd also like to finally thank my fellow speakers for, I think, they delivered some brilliant molecular biology accessible by surgeons. Thank you very much indeed.